So this is a tutorial on sex-linked genetics problems. In animals, here genes linked to sex determination can be found on specific pair of chromosomes. For example, in humans and in some insects, for example, the pair of chromosomes determine sex are the XY chromosomes, which in our karyotype would be the last pair. All the other pairs of chromosomes, the other 22 pairs, are referred to as autosomes, and they're homologous. So that means they carry the same types of genes. There just may be different alleles for each pair of genes. But when it comes to the sex-determining chromosomes, the two chromosomes are different. In humans, the Y chromosome is much smaller and it's missing many of the genes that we find on the X chromosome. So the two chromosomes are the sex chromosomes. They're not homologous exactly because we're missing many of the genes. So there's maybe no more than 30 genes on the Y chromosome, whereas the X has over 1,000 different gene loci, or over 1,000 different genes. But in humans, if you get an X, two X chromosomes, you would be female, and if you got an XY, you would be male. So that's why they're sex determining or sex chromosomes. Now, if another trait occurs on any of these chromosomes, then that trait is also linked to the sex of the individual. For example, if there is a gene locus on the X, then we have a sex linkage to the X chromosomes. We would refer to it as X-linked chromosomes. So that would be our definition for sex linkage, is when another trait is determined by a gene and is also located on a sex-determining chromosome, then this trait is referred to as sex-linked. So you can see other animals have different ways, different systems for determining sex. So pedigrees can be used to show inheritance patterns, especially for humans when we look at family histories. And so that's what a pedigree does, is it tracks them. And any one individual that is shaded in would be carrying the trait. So here a square would be a male carrying a trait, and a circle would be a female carrying a trait. If the square or circle is open, then for example this would be a female that's not carrying a trait. But we're going to focus on the ones on the bottom at these pedigrees. And when we look at these pedigrees, they overall can be used to suggest a certain inheritance pattern for a trait that we're trying to track. And these are some of the patterns. For example, there's autosomal dominant, which just means the trait is carried on a regular autosome rather than the sex chromosomes. We have autosomal recessive, where you see neither parent carries, so it appears to skip at least one parent generation, but then you see the trait appear in the next one. That's characteristic of a recessive autosome. And then we have X-linked inheritance patterns, and then X-linked dominant and X-linked recessive. So if we look at the X-linked recessive, you see neither parent shows, but since the gene is carried on the X-linked chromosome, a male is only going to get one copy. So you're going to have overall a clue that this might be, because you can't prove it, that this is an X-linked recessive, is when you tend to see only males, or a lot more males than, and rarely any females that show the trait. So that would be suggestive of X-linked recessive disorder, where we see only sons are affected. So that would be sort of answering this last question here. What pattern would suggest a recessive X-linked? Well, here you're going to tend to see, you can see the trait skip a generation, but you're going to have a high incidence of overall more sons and rarely any daughters that inherit this trait. So let's take a look at our first X-linked recessive inheritance problem. And so here we ask, what are the probabilities of having a child with color blindness, which is a recessive X-linked trait? And so here we have a normal father and a mother that's a carrier of the colorblind trait. So we need to define some alleles, and we're going to do so by actually keeping track of the X and the Y chromosomes as well. So we're going to use X for X chromosome, and the X would have a capital N for, we're going to define that as normal vision, and capital because the normal vision is dominant and colorblindness is found to be recessive. So this would be normal vision, and a female, or I'm sorry, not necessarily female, but X chromosome with a little n at that location would be the mutated allele for color blindness. So this would be colorblind. And then Y does not have that allele at all because it's a very reduced chromosome. So that would be your just keeping track of the Y chromosome. So here we need to first look at trying to figure out what are the genotypes here using these as our symbols for alleles. We know that a father, male, has to be XY. Okay, and this is a normal father, so the father is actually only carrying one copy because the Y doesn't have that gene location to begin with, or gene locus. The female has both X chromosomes. That's what makes a female, but this is a carrier. So she's not colorblind, but she is carrying one of the X chromosomes is carrying the colorblind allele. So now we have to do is set up a Punnett square, as we've done before. Put one of the parents on top and the other parent on the left side of the Punnett square as far as the types of gametes they can make. Now during meiosis, we're basically going to be splitting the X and the Y chromosomes. So half for the male are going to be, half of the sperm produced are going to be carrying the X chromosome, and in this case the X chromosome is carrying a normal allele. The other half are going to be carrying the Y. And this is actually why the male determines the sex. So overall, you have to get a Y, and the only way you can get a Y is from a male or from the father. And so the female has two X chromosomes, but in this case, this female is a carrier for color blindness. So now all we do is we go in and fill in the Punnett square all the possibilities. Remember, if we assume random fertilization, then we're equally likely for a sperm from a male carrying the normal allele for vision to fertilize any of these eggs. We're also equally likely for sperm, because half of them are carrying the Y chromosome, to fertilize any of the possible eggs from the females. So what we're going to get is in this box here, 1/4, 1 out of 4 boxes are going to be homozygous normal female, and then one box where we're going to have a male with normal vision, and there's no such thing as homozygous, heterozygous, because you only get one copy here. And then over here we have our carrier, and she would have normal vision because she at least has one dominant allele. And then over here you're going to have a male with only one chance or one copy of that gene, and in this case it is the mutated colorblind gene. So the question asks, what are the probabilities of having a child with color blindness? The probability is only going to be 1/4, and in this case all of them are going to be males. So the answer here would be 1/4, not 1/2, 1/4. You can also answer this as a percent, 25%. So 25% of the offspring are going to be colorblind and male. There is no chance at all for any females to be colorblind in this case here. And this demonstrates why when we see an X-linked recessive pattern, we have a tendency to see more males than females. Now, how is it possible? Is it possible to get females that are colorblind? Yes, but we would have to have a female that's a carrier, and we got a carrier right here in this example here of this genetics cross. So we got a carrier right here. Okay, and so let's say this individual is born and is not colorblind, but she would be carrying, female would be carrying the allele, and the female carrying the allele would have to basically have children with a male who's colorblind. Because if the male were not colorblind, then he would have a normal X chromosome, and remember, each one needs to give one or the other chromosome. So if the male had normal vision, then there would be no way, because all the offspring from a normal male would always get at least one normal copy. So the condition in order to get a female colorblind, you would have to have a male who's colorblind and at least a female who's a carrier. And if you went and you worked out the Punnett square, then it's possible to get a female. It would actually be 1/4 of the 1 out of the 4 boxes in the Punnett square would show females that would be carrying both, and that would make her colorblind. So remember, there would be a half chance of getting this particular chromosome, and then a half chance of getting this chromosome to be female. So there's a half chance of being female, but there would be a quarter chance of all the offspring would give us this. So if a couple were to have children that had this genotypes that in this example right here, okay, if they were to have that, then they would there's a quarter chance that you would have both female and colorblind in that case. In the next example, we're going to look at fruit flies, and in fruit flies they have a similar sex determining system with an X and Y chromosome determining sex. So both X's would be female and XY would be male, just as we see in humans. And so we need to look at defining our alleles as before. So fruit flies, it is shown that there's a mutation for white eye color, and it's actually a recessive X-linked trait. So we would see a similar pattern where you would have tend to have more males than females if you followed a generation of genetic crosses with them. Although it is, again, more rare but possible to get females with white eyes. So here, the normal color is going to be red. So the question here says, if a female carrier for a white-eyed trait is crossed with a white-eyed male, what are their probabilities for the offspring? So let's define. It's on the X chromosome, and the dominant is red, so we're going to put a superscript of big R, so this would be red eyes. And then little r for the recessive white eyes, and then Y for the Y chromosome. So going back and looking at the problem again, we have a female carrier. So we're going to set up our cross or symbolize it. So the female fruit fly would be a carrier. So that means there's one red eye allele and one white eye recessive allele. And the male is white eyes. Okay. So the male has only one copy, and it's for white. And this would be a white-eyed male. And this is a case similar to the hypothetical example I gave earlier, where you can get a colorblind female. That would be the case where the female has to be carrying at least one of the alleles, and the male has to have the character or the trait. So running the Punnett square here, we'll put the female fruit fly on top and the male fruit fly on the side. They're possible gametes, and then we go in and we fill in, and then we can figure out what the probabilities of having the certain types of offspring are. So this one would be red vision male, or not red vision, but red-eyed male, and this would be a white-eyed male. So you can see here, you're actually going to get half of the offspring are going to have white eyes, and the other half on the left side over here are going to be red-eyed. The female would be red-eyed carrier. But of the offspring that are white-eyed, half of those white eyes are going to be female, and the other half would be male. So overall, if we look here, what are the probabilities here for all of the offspring, for the possible offspring? If we were to answer this appropriately, we would say, well, in a genetic crossing with two parent fruit flies with those genotypes that are defined here, then 1/4 of the offspring, or 25%, you can answer either way, 1/4 or 25% would be red eyes and male, female for the first box. 1/4 would be red eyes and male, which is the bottom left box. Then we would have 1/4 chance of getting, or 1 out of 4 chance of getting white-eyed females. And the last box would be 1/4, or you can write 25% if you want, would be white eye and male. So this would be another example of a genetics problem where you have a female carrier and a male who has the recessive trait, X-linked trait. So now we look at going back to these pedigrees, and we see two types of patterns. The two problems I gave you were showing X-linked recessive, and if this were the case in humans, we might use pedigrees for the analysis. So the bottom or the right picture here of the pedigree, this would be generation 1 and this would be generation 2. This would be individual 1 and 2 in the first generation, and then 1, 2, 3, 4, 5, and 6 for the second generation. So overall they would be, if we look at this interpretation here, the circle is the female, not showing the trait, male not showing the trait, so that tells me that it's probably recessive. And then when we look at all the children, only the males have it. So this pattern is starting to suggest that this is X-linked recessive trait. Because we'll tend to see more males that will have the effect, since they only get one copy on the X chromosome. And so the idea here would be to go ahead and determine what the genotypes are. So if we start looking, and this is X-linked, okay, then I know that a male, for example, individual 1, number 2, which is male, only has one X chromosome and does not have the condition. So we're going to use capital N for normal trait, okay? And then Y. That would have to be the genotype for this individual here. For the female in the first generation, the mother there, then the only way to have children to have it would be, and it's recessive again, is if she's carried. If she was not carried, then there's no possible way to get any offspring that have it. So these, we know for sure what those genotypes are for those individuals. We also know that because these are males and they're showing the condition, then individuals in generation 2, individuals numbers 3 through 5, the shaded N boxes, have to have the copy of the recessive allele, and that would be their genotypes right there. Without question, you know that, okay? The only other one I think that we would know for sure would be the male, the first male in the second generation, because the male does not have the trait, is not, whatever that recessive trait is, does not have it, and it's recessive. If he's not showing it, then he has to have, since he only has one copy, would be the dominant trait, and that would be his genotype. As far as the females, number 2 right here and number 6 in the second generation, it would be impossible without knowing what kind of children they would have later if they grow up and have children in the next generation. It would be impossible to say with any degree of certainty what their genotypes are. We know that they are showing the dominant trait and it's X-linked, but we don't know whether or not they're carriers or whether or not they are have both dominant. So the best we can say is that they have one dominant allele for whatever that trait is, and the other one we don't know. Okay, so that would give us females that don't show the trait, so they can be either little n or big N in that position. So that's it for this tutorial on X-linked traits. I hope it helps.